Thank you. So yeah, my name is Xena Dyball. I'm working at Public Health England Portadown in the Technology Development Group. And I'm just going to be presenting some work that I did as part of my master's project over the past year here at Public Health England. So the work I've been doing is on characterization of phage susceptible and resistant isolates of Acinetobacter baumannii using a novel impedance cytometer. So just a little bit of background behind my work. As I'm sure many of you are aware, there's been a recent resurgence of interest surrounding the practice of phage therapy due to its potential as a alternative therapeutic approach to treat infections caused by multi-drug resistant bacteria. And although there are many benefits to phage therapy, one of its limitations is the kind of initial phage selection process, which requires the use of susceptibility tests in order to assess bacterial susceptibility to phage infection. This process is important as it ensures that only the most effective phages are selected for patient treatment. However, traditional phage susceptibility tests that are commonly used, such as plaque assays, spot plates, and broth microdilution assays, are relatively time-consuming and slow, producing results in roughly 16 hours or longer, which subsequently increases the time a patient could be waiting for treatment. And could ultimately impact their overall outcome, especially if a patient is kind of coming in for phage therapy as kind of last resort treatment and they're failing kind of traditional antibiotic therapy. So therefore, there is a need for diagnostic tools that allow bacterial susceptibility to phage infection to be determined more rapidly. So one rapid susceptibility test that has been developed by a group at the University of Southampton is the iFast platform. And iFast stands for impedance-based fast antimicrobial susceptibility test. And this platform uses microfluidic impedance cytometry to determine bacterial susceptibility to antimicrobial agents, so things such as antibiotics and, in my case, phage. So this technique functions by using micro electrodes to measure the electrical signatures of single cells and particles as they flow through a microfluidic chip, which is just shown here. So these electrical signatures can be recorded as the cell's impedance value, and then they can be used to characterize subtle biophysical changes that occur in the cells, enabling the effects of antibiotic or antimicrobial exposure to be measured after a very short time window. So antimicrobial susceptibility in bacteria can be assessed really quite rapidly. So in the paper published by the group from Southampton, they demonstrate susceptibility in bacteria to different classes of antibiotics within 30 minutes, which is obviously extremely fast. If you are interested in more about how kind of iFast functions and kind of the electrical engineering behind it, I would really recommend reading the paper, which I've just linked here, as they also go into a lot more detail than I have here in the talk today. So this is just an example of the kind of results that we generate from performing experiments on the iFast. So in this experiment here, the bacteria Klebsiella pneumoniae has been exposed to the carbapenem antibiotic meropenem for 30 minutes at its clinical breakpoint. And on the scatter plots here, the x-axis represents the electrical radius of the particles or cells, and then the y-axis represents the membrane permittivity, or sometimes we refer to it as the opacity of the cells and particles. So essentially, each dot on the scatter plot represents a single cell or particle as a measure of its electrical radius and kind of conductivity. So this red gate here represents the kind of untreated initial cell population. And then when this contour is applied to the scatter plot on the right here, which is of bacteria post-antibiotic treatment, it highlights the morphological changes that have occurred in the cells as a result of meropenem. So you can see that in this plot here, the population has completely moved out of the contour from the control sample here. And this movement can be broken down and linked directly with how meropenem functions in susceptible bacteria. So the way how we think meropenem functions is that it breaks down the cell wall of susceptible bacteria, which then causes the cells to swell in size. So we can interpret that on this plot here. So the breakdown of the cell wall can be interpreted as the reduction on the y-axis here. So the cells have moved down out of the contour, so reducing in membrane permittivity. They're becoming less conductive because the cell wall is breaking down. And then the swelling of the bacterium size can be seen as the shift on the x-axis, then moving along the x-axis, increasing in electrical radius. So this just demonstrates basically how the movement of cells out of the contour following antimicrobial treatment can be used to characterize bacterial susceptibility. So far, the iFAST has only really been used to assess antibiotic susceptibility in bacteria. So the work I'm presenting today is kind of the first time phage susceptibility has really been investigated on the iFAST. So I've just got a diagram here of the kind of protocol I developed to perform my phage susceptibility experiments on the iFAST. So to run these experiments, I would inoculate my bacterial culture with phage at either an MOI of 5, 1 or 0.01. I also had an untreated control sample. I then incubate these inoculated samples statically at 37 degrees for 2 hours. And then following incubation, I would then read them on the iFAST for approximately 2 minutes per sample. So overall, this susceptibility experiment takes a little over 2 hours to kind of set up, run and generate results. So the raw data that we get from the iFAST is these scatterplots, which you've seen on the previous slide. And they take a little bit of analysis in order to be able to interpret them correctly. So analysis of the plots is performed using MATLAB software. And this allows us to draw a gate around the untreated initial cell population on the control sample. And then from the gate that we've drawn, the software is then able to apply that to the treated samples. And then it can calculate the total cell counts within the contour, and then also the mean electrical radius of the cells on the plots. And then we can use this information to kind of quantitatively assess whether a strain is susceptible or resistant to phage infection. So for the experiments on the iFAST, I chose to use Xena Dyball mannii as the target bacteria. So this species was chosen as it is a WHO priority pathogen for which new antimicrobials are urgently required. Furthermore, multi and pan-drug resistant strains have also been isolated and they've been observed to cause a variety of infections in critically ill clinical patients. Additionally, the CDC has estimated that carbapenem-resistant A-bioweaver mannii caused approximately 8,500 cases in hospitalized patients and 700 deaths in 2017. So therefore, patients suffering from multi-drug resistant Acinetobacter mannii infections could really benefit from phage therapy and rapid phage susceptibility tests such as the iFAST. So in order to target the A-bioweaver mannii, I used two different Acinetobacter phages. So these are two lytic phages called Loki and Freya, and they were kindly given to us by Dan Turner from the University of the West of England. So the host strain for Loki is ATCC17978, and then the host for Freya is NCTC13302. And these were the phage and strain combinations I used for all of my experiments, which I'm now about to show you the results for. So these are just the results generated from my experiments on the iFAST with Loki. So here I used the susceptible host strain 17978, which is highlighted in green. And then I also used a Loki resistant isolate of 17978, which is this colistin resistant 17978, which is highlighted in red. And this isolate basically has a mutation in the LPXA gene, which affects LPS synthesis. And it's been hypothesized that Loki uses kind of a component in LPS as the host cell receptor. So that's what makes it resistant, because it's lacking the LPS. So at the bottom of the slide here, we have the iFAST generated scatter plots, and then above are some bar charts showing the compiled iFAST generated data of 3 repeats for both of the strains with Loki and MOI of 5, 1, and then 0.01. So the bar charts clearly show that there are significant decreases for all 3 MOIs tested in the susceptible host strain compared to the untreated control group. Whereas with this Loki resistant isolate, we only see kind of significant decreases in the percentage of cells in contour at MOI of 5, and then again slightly at MOI of 1. But this kind of shows that this isolate is more resistant to Loki infection than this host strain here. So the scatter plots are a little bit more difficult to kind of interpret, but basically what we're seeing is that with 17978 here, we see a movement of cells out of the contour kind of shifting towards the right, so increasing in electrical radius compared to the untreated control group here. And then with the loci resistant isolate with MR of 1 and 0.01, and these populations are looking very similar to the untreated control group, kind of suggesting that cells aren't moving and they're resistant to loci infection. At MR of 5, we don't see a movement of cells as such, we kind of just see a reduction in the overall number of cells present. But obviously we see that in the bar chart here as well. So similarly to the last slide, these are the results for the experiments performed with the phage Freya. So I used the susceptible host strain 13302, and then also a Freya resistant isolate of 13302 called PA1. And this isolate has a mutate or an insertion transposon mutation in PGLC gene, which affects kind of the capsule biosynthesis pathway. So we're assuming that Freya uses a component of the capsule as the receptor, and that's what makes this one resistant because it doesn't have the capsule. So again, with the bar charts here, we see that with the host strain at all 3 MOIs tested, there are significant decreases in the percentage of cells in contour compared to the untreated control group. And then with the Freya resistant isolate, we only really see significant decreases at MOR 5, and then at 1 and 0.01, these populations are remaining very stable and similar to the untreated control group, showing that this isolate is more resistant to Freya infection than the host strain. And we see that in these scatter plots here. So with the susceptible strain, unlike loci, we don't really see a shift of cells. We just see a kind of overall reduction in the number of cells present for all 3 of the MOIs tested. And then with the Freya resistant isolate, we only see a kind of reduction in the total number of cells at MOI 5, but that's represented here in the bar chart as well. So just as a comparison to the results generated by the iFast, these are some growth curves which were produced by performing broth microdilution assays with the various ABAM and AI isolates and loci and Freya. So as you can see, these growth curves show very similar susceptibility and kind of resistance profiles to those produced from the iFast. So the 2 susceptible strains here show substantial decreases in the levels of growth or optical density in comparison to the untreated control group, which is the red line. And then with the 2 resistant isolates, the phage treated groups all kind of show very similar growth patterns to the untreated control group. There is a slight reduction in growth at MR 5 in this strain, and then 5 and 1 in this strain here. But again, that matches what we saw from the iFast. So overall, the results produced by the iFast match those generated by traditional susceptibility testing methods, but they're produced in a fraction of the time required by the traditional methods. So just to conclude, from the results shown here, the iFast can detect phage infection in bacteria within 2 hours and can also differentiate susceptible and resistant isolates. The iFast platform therefore demonstrates potential as a rapid phage susceptibility test and could potentially be used in the future to kind of aid the initial phage selection process. However, obviously more work needs to be done in order to assess the iFast full range of applications, as it still is in its very initial early testing phase. But maybe future work could include stuff like assessing host ranges of phages and looking at real-time resistance emergence, also looking at assessing phage cocktails and kind of phage synergy with antibiotics also. So I'd just like to round off by saying that the work on the iFast that I've presented here today is linked with my group's Open Innovation in AMR model, which is open to scientists from all backgrounds to come to Public Health England Portendine. While here, they'll receive training in microbiology techniques which are relevant to early stage antimicrobial research. So we have an established screening cascade for the evaluation of novel compounds, which is just shown here. And this screen includes non-traditional therapeutic approaches as well as small molecules. Additionally, we also have a range of facilities such as a large clinical strain library, whole genome sequencing, and the hollow fiber model for PK/PD studies. We also have a fully functioning modular hospital wards at Portendine where we can test infection control and prevention measures, also study disease transmission and assess infections. So if anyone has any questions about the model or are interested in discussing their projects with us, the contact email is just here. So it's amr.screening@phe.gov.uk. And then finally, I'd just like to say thank you to everyone who made this work possible, and particularly Dan, Howell and Roll from Southampton for providing us with the iFast, Mark and Charlie, my line managers, and then also Dan Turner for providing us with the phages. And thank you for your attention.